Triskel is a biomedical company which works closely with the University of Groningen. Triskel designs, produces and tests improved variants of trail, a human protein, involved in so-called programmed cell death, apoptosis. Apoptosis is a naturally occurring process that's involved in the elimination of diseased cells.
Trail binds to receptors on the cell surface, so molecules that are expressed on the cell surface of these cancer cells, but also normal cells. And it's capable of binding to 5 different types of receptors, and only 2 of these are capable to induce a signal of this programmed cell death and thereby eliminating the cell.
Johanna Vrielink is a biochemist working in the pharmaceutical department at the University of Groningen. She produces and purifies the receptors to which the trail protein can bind itself. Once she knows what they look like, she can change trail so that the protein only binds to the 2 receptors which play a role in cell death. In order to study these receptors properly, she cultivates them in bacteria cells.
We can express these receptors in the bacteria cells. To do this on a large scale, we use the fermenter. The fermenter is an apparatus where we can, in one time, make 5 liters of bacterial cells. And afterwards we select the cells and we break them. And then from all these proteins we purify only the protein of interest and separate it from all the other bacterial proteins.
If we can bring in specificity into the trail interaction with the receptors, just by introducing a couple of mutations and thereby eliminating in fact the interaction with the decoy receptors, we can make the apoptotic signal more efficient.
The main goal is to purify the decoy receptors of trail and to get a pure and homogeneous sample of them, so that we can crystallize to make 3D structures of these receptors, and then we can gain more knowledge about these receptors and with that also of the function of trail.
Once Johanna Vrielink has a good picture of the structure of the receptors, new trail variants will be designed using a computer model. In the end, these new trail variants will be tested with various techniques to find out whether these make good contact with the receptors they need to contact.
The final aim is, of course, to develop a therapy for cancer patients that is efficient. What we hope is to develop a sort of blockbuster where we can develop therapies for all types of cancer.